Good morning, good afternoon, and good evening to everyone. Thank you for joining the symposium and thank you for joining today's talk. Unfortunately, I'm really sorry, but I can't be there in person. Unfortunately, the way time zones work, it's not so great, but I hope that this video presentation will be interesting and exciting to you. And of course, at the very end, I'm happy to take questions by text, by email, by chat, or any other way. So today I'll be talking about the origin and evolution of biopolymer and non-biopolymer based compartments. So first of all, I'd like to start with a little bit of a self-introduction. So I actually did my bachelor's degree at Caltech where I studied chemistry and business economics and management. And part of my research involved studying chloroplast protein expression in crystallography, as well as using mass spectrometry to study pulmonary surfactant lipids. Next, I moved to Harvard where I did my PhD and I studied mostly RNA. I studied RNA sequencing technologies, specifically used for non-coding RNA quantification. And I also studied ways which RNA could polymerize and replicate without using enzymes, so-called non-enzymatic RNA polymerization. Because I had so much experience working with all of these different biomolecules, lipids, proteins, nucleic acids, I got really interested in how exactly those biomolecules emerged and evolved in the first place. And that led me to my current position. So, since 2017, I've been a researcher at the Earth Life Science Institute at Tokyo Tech Institute of Technology. And also, since 2019, I've been an affiliated scientist at Blue Marble Space Institute of Science. And since then, at ELSI, I've been working on a variety of different topics related to origin of life and astrobiology, but also related to other interesting topics such as DNA liquid crystals or continuing with some RNA sequencing technology work. So before I get into today's main part of the talk, I'm going to turn off my camera in case there's if in case the portrait interferes or blocks some of the content. So today I'd like to go through four different, minor topics. So in case you aren't familiar with what exactly is astrobiology, especially in the eyes of a prebiotic chemist, I want to introduce and give maybe a little bit of a review about primitive cells and what they might look like, and then give two short case studies in origins of life research featuring different polymers. One is from a biopolymer based background, one is from a non-biopolymer based background. So hopefully you can see the similarities and differences of those sequences and those systems that we study. And finally, I want to introduce some opportunities for students and researchers to participate in astrobiology research, both at ELSI, my current affiliation, as well as outside of ELSI. So, what exactly is astrobiology? I think many people have different concepts of what exactly is astrobiology. For example, if you take a look at the word astrobiology, you would think that it's the intersection of astronomy and life science. And basically, it is. Astrobiology takes, concepts from astronomy, takes concepts from life science, and what astrobiology is encompasses many different things. So, what I think, and from a prebiotic chemist perspective, what I think astrobiology research encompasses includes, for example, the active search for life in space. This is, searching for current life or former life, and often this is through missions. For example, in Japan, there's a Hayabusa 2 mission which went to get samples from the asteroid called Ryugu. It recently returned to Earth a couple years ago, and they're studying those samples right now. And in a few years, JAXA will be sending a mission to one of the Martian moons and to study kind of the structure of the Martian moons and bring those samples back. So that's one aspect. Another aspect is searching for habitable planets in space. So, for example, searching for exoplanets, probing the habitability of extraterrestrial bodies, and this can be done, for example, through space-based telescopes like the James Webb Space Telescope shown on the left, or through these radio telescopes that are on Earth, and this is ALMA on the right. Next, we also have the active search for the origin of planets, Earth, and life on Earth. And so this includes the search for organisms that live in extreme environments, which might provide a window into what life was like back on early Earth, as well as research on planetary and geological formation. And this also involves studying, areas of the Earth which might simulate areas that one could find on other planets. And these three last topics are something my research has a hand in. So the search for how to make synthetic living systems in the lab, how primitive chemicals assembled into primitive life, and finally how this primitive life evolved into modern life. So this is the topics that I'm personally researching. It's part of this subfield of astrobiology, which is the origins of life field, and it's a really small part of the field, but extensive collaboration is really essential in order to really understand aspects of the origin of life, and especially collaboration with different types of researchers, and even researchers or those with an engineering background or people from non-scientific backgrounds is really essential. So coming from kind of a biochemistry or a biology background, I'm sure many of you are really interested in how the cell works, what the structure is. And so, this is a simplistic cartoon of the cell, but essentially it's built up of many complex parts, and researchers have really had a lot of success understanding many of the structures and what they do and what they're made to do, or maybe during diseases when they have trouble. And we've learned a lot about the cell in the last 20, 30, 50 years, but there's still many unanswered questions. And one of these big unanswered questions is, what did the first cells look like? How did these first cells function? And some researchers believe that something like a more primitive type cell existed on early Earth. Some people call this a protocell. And maybe this included some type of compartment boundary, some type of replicating genetic system, housed, catalysts like enzymes or primitive enzymes, and then also could participate in evolution, growth, and division. And so one way to think about origins of life research is we want to understand how the chemicals on early Earth, in the prebiotic soup, if you will, assembled into these primitive compartments or protocells, and how these protocells functioned and eventually evolved into modern cells. And specifically, if we think about this, a non-biomolecule or non-biopolymer-based prebiotic soup, how could this eventually assemble and evolve into a biomolecule or a biopolymer-based modern cell is a really interesting and big question. And in order to do this, one way is to try to see or understand what this primitive cell looked like, what it could do. And we can approach this in two ways, going from the bottom up, building a primitive compartment using these non-biomolecules available on early Earth through prebiotic chemistry, or we can go from the top down, taking what we know about biology and kind of working backwards to see what minimized functions or what minimized types of cells could have existed. So what I like to say is we want to build this primitive compartment from both directions. And so let's say going from top down, I think this is something that more of you in this audience are familiar with. Let's see how this primitive cell would have looked like if we work backwards from a modern cell. And forgive the simplicity of this cartoon as well. This is one of the processes we see in the modern cell. It's, as you know, the central dogma. And these are composed of different processes made up of biopolymers and biomolecules and catalyzed by biomolecules. So what we want to know is on early Earth or in an early cell, which of these processes were still essential. So, for example, it's very likely that proteins did not exist on early Earth. They're too large. Their folding is too difficult to have achieved. And also, it's very likely that a DNA couldn't have existed really on an early Earth. So this leads many to postulate that the first cells on early Earth contain RNA. And this is specifically leads to a origin of life hypothesis, which suggests that RNA is the first and most important biomolecule of life. And this hypothesis is called the RNA world hypothesis, and it postulates that because RNA can store and pass down genetic information, because it's similar in structure to DNA, and also because RNA can fold into specific structures that can catalyze chemical processes in the form of ribozymes, similar to proteins, that RNA was the first and most important biomolecule on early Earth. And while there's been a lot of research in this field and a lot of progress has been made, there's still a number of outstanding questions in the RNA world. For example, how the RNA could be synthesized, how it polymerizes, how it's prevented from hydrolyzing. There's many questions that researchers are answering to this day. And there are also hypotheses that RNA wasn't the only biomolecule that existed on early Earth. For example, some researchers have postulated that DNA and RNA could have coexisted on early Earth, maybe in a hybrid form. And based on the structure of the ribosome as we know it today, it's also been postulated that perhaps RNA and peptides co-evolved. So instead of a purely RNA world, you might have had something like an RNA peptide or an RNA protein world. And so what all of this is just saying is that we're not exactly sure exactly what was in this early cell. And it's plausible that all of these or even more types of molecules existed in or around these cells. So let's assume from now on that we have access to a number of different types of biomolecules for building this primitive cell. The next thing I want to focus on is this boundary. So this boundary is really important and it defines a cell from other cells. Specifically, it makes the cell defined as a compartment. And compartments are really essential to origins of life. Compartments segregate and concentrate reactants. They promote chemical reactions, promote evolution, prevent fusion of genetic materials, many different important functions on early Earth. And modern cells, they're made up mostly of a phospholipid membrane. That's the boundary. It's been postulated that perhaps early cells that are membrane bound were made up of fatty acids rather than phospholipids because they're much easier to synthesize using prebiotic chemistry and what's available back on early Earth due to there being only one chain rather than two. And so one further question is that can you actually make a boundary that's even simpler than fatty acids that doesn't even require that type of bilayer? And so what we're interested in is seeing whether such types of compartments could have existed and whether they could form or function as early cells or protocells. So one way is through liquid-liquid phase separation. For those who are familiar with this, you might have heard about this. If you haven't, it's actually one way to form droplets in solution. These droplets are membraneless, but they're still liquid, and they provide compartmentalization. Without a membrane, they allow exchange of molecules between phases, and in fact, they're present in modern biology. So there's a phenomenon called biological phase separation, which results in formation of membraneless organelles in the cell. These organelles, there's some of them are called, for example, nuage granules or stress granules. Maybe chromatin is even considered one of these, and these are membraneless organelles in the cell that are really important for controlling biological processes. And it's been postulated that because most cells or almost all cells have this type of phase separation inside to make these membraneless organelles, that perhaps they're an ancient invention and that phase separation was also used to make compartments on early Earth. And so here, let me move on to a couple case studies of origins of life research, and let me start with the biopolymer case. So this is a case study on primitive liquid crystal coacervates. So you might have heard of liquid crystals before. They're present in TVs or displays. And in fact, you can actually make liquid crystals out of DNA. So this is in collaboration with Tommaso Fraccia, who's a researcher at ESPCI in Paris. And so his previous research showed that if you took short DNA oligomers that were self-complementary, such as this sequence shown here, they can actually form these duplexes that aggregate end to end. They stack together and they form these long rods. And these rods will bundle together, and after drying, after increasing the concentration of the DNA significantly, the structure that results is this columnar liquid crystal structure, shown in this polarization microscopy image on the right. So these structures are, somewhat liquid. They have liquid and solid characteristics, and you can visualize them through polarization microscopy because of their supramolecular chirality. So we postulated that perhaps it would be interesting if, for example, a primitive compartment, like one of those phase-separated systems, could actually house one of these liquid crystal structures because they've shown the ability to affect electrochemistry or regiochemistry or regioselectivity. So what we did was we knew that, when nucleic acids like DNA and when peptides, like cationic peptides, are mixed and bind together, they actually can form these liquid-liquid phase-separated structures. So we took PLL, polylysine, about 250 residues long, polydisperse, mixed it with high concentrations of this DNA oligomer. And we were able to find conditions in which these structures, these nice membraneless droplets or coacervates, co-localize with this DNA crystal, liquid crystal structure shown on the bottom through the polarization microscopy. So we believe that this DNA liquid crystal structure is formed through this end-to-end stacking interaction of DNA. And we actually found that these structures, the co-localized structure, the liquid crystal coacervate, only exists in a very narrow range of salinity. For example, when we varied the salinity, we found that at low concentrations of sodium chloride, precipitation occurred, whereas at higher concentrations of sodium chloride, only droplets formed without liquid crystal characteristics, or no droplets could form at all. And we believe that this is because the ionic strength of the solution is really important for this interaction. If there's lower amounts of salt, the peptide DNA binding interaction may be too strong, forming precipitation or solid-like particles. Whereas at higher salt concentrations, it makes these structures, these interactions between the peptide and the DNA weaker, suggesting that when the peptide and DNA can no longer interact, for example, at 1 molar sodium chloride, these droplets can't form as well. We next wanted to test our hypothesis as to whether this end-to-end interaction is indeed what's forming these liquid crystal structures. So we did the same experiment, but with a slightly different DNA sequence. This time we included a 2T base overhang so that this stacking process could not occur. And we found that through all of the different conditions we tested, in no single case did we find liquid crystal structures. We did find many conditions where these droplets could form, these phase-separated droplets could form, but liquid crystals could not be observed. And this shows that this end-to-end stacking interaction is really essential for these liquid crystals to form. We then next wanted to study the what happens if we subjected these liquid crystal coacervates to processes or environmental processes that might have existed on earlier. So one of them is dehydration. It's been proposed that dehydration could be one way to effect different chemical reactions on earlier. You'll see this in the second part as well. And so we subjected these systems to drying. And so what you see here is that throughout the drying process, you can see these structures go from these solid-like structures transitioning to these liquid crystal coacervate-like structures to just a simple coacervate and then to a disassembled structure. I'll play this video again. And we believe this is the case because, similar to the salinity titration, dehydrating the solution actually up-concentrates the sodium chloride in the solution, perhaps, which suggests that this phase transition follows a similar phase transition as what we observed in the sodium chloride titration case. Next, we started again and we wanted to see the reaction of when we heated these samples. So we actually heated these samples up to about 60 degrees and we can see a transition from this solid-like structure to a liquid crystal coacervate structure. Furthermore, further heating resulted in disassembly of these liquid crystal structures, resulting only in these non-liquid crystal coacervates. And then upon decreasing the temperature, we noticed that at 40 degrees on the way down, these liquid crystal coacervates started to pop up again. And we saw a few different types of liquid crystal coacervates, but also we saw that the population was also heterogeneous. Some of them were still typical droplets without liquid crystal characteristics. Some of them had liquid crystals inside. And we found that upon decreasing at further to about 10 degrees that almost all of these droplets reconstituted this liquid crystal character. If we compare between these before and after heating, the structures are pretty different. And we think that this is because this heating process is basically akin to an annealing process. And of course, annealing is used to allow in DNA and RNA case to allow the strands to bind completely to find the most stable thermodynamic conformation. And we believe that in this case, perhaps the right figure here is this is what the most stable conformation is for this system. And so we believe that what's happening is we have these liquid crystal structures here with increasing heat and salinity. You have these long DNA rods in teal, and these red strands of peptides bound together. The DNA rods are in this packed hexagonal pattern. Upon increasing heat and salinity, these rods start to come apart, causing some structural change. And then the rods start to disassemble these into, destack. And so you lose this liquid crystal characteristic, but you retain the droplets. And then further increasing heat and salinity results in disassembly of everything, and you result in no droplets. So for those who are interested in learning more details, some of these studies are published. You can see one here and one here as well. I'd highly encourage you to take a look if you're interested in the details. Next, I'd like to talk about our other side, non-biopolymer side of origins of life research. And here we focus on membraneless polyester droplets. So here we're building from the bottom up, and you see we're interested in building this primitive compartment from the available chemicals on early Earth. And it's actually known that there's many ways by which biomolecules could have formed on early Earth, through meteorites or atmospheric discharge. But we also find that together with biomolecules, that there's also many non-biomolecules synthesized at the same time. For example, Murchison meteorite has tons and tons of molecules inside. And if you analyze a Miller-Urey reaction or atmospheric discharge reaction products, you also get tons and tons of different molecules. Not all of these are biomolecules. This suggests that non-biomolecules were just as or even more abundant than biomolecules on early Earth. So we're interested in understanding how non-biomolecules could have catalyzed life's emergence. The system we like to use are alpha hydroxy acids. They're monomers similar in structure to amino acids, but with an amine group that's replaced by a hydroxyl group. And what's great about these is that through simple drying, these monomers can form polyester polymers, shown on the bottom here. So we wanted to create these polymers, these polyesters, and see if they could, assemble into something useful for the origin of life. So we first took monomer solutions and dried them to form polyesters. And here's some mass spec data, and you can see that in all cases, the monomers were able to polymerize into polymers. Let's zoom in again. This is one of the monomers, lactic acid. So you see that the products are polydisperse. They're not a single product. So you make many different lengths of polyesters. And so you also form this gel-like structure. So this was observed in almost all cases where all of the polymer gel structures were formed through by these polydisperse polyester products. So we took these gels and we dissolved them in aqueous solution. And what we found was that droplets could form. They were turbid, and we could see these nice spherical droplets in solution for almost all of these structures. We actually took these droplets and we assayed them a bit further. So we first found that these droplets could, encapsulate functional superfold GFP. We also found that these droplets could catalyze or scaffold the assembly of some type of lipid structures around the droplets. And finally, we found that RNA could segregate into and function in the presence of these droplets. So one of the next questions we tried to pursue was that, only one type of polyester droplet we found could compartmentalize RNA, but these are all neutral droplets that we studied. Can other polyester droplets or droplets made from other compositions compartmentalize RNA? And we hypothesized that if we included a basic alpha hydroxy acid, something that could be positively charged in solution, the droplets that contain this positive residue could allow the RNA with a negative backbone to segregate more strongly than before. And indeed, when we incorporated some percentage of these cationic residues, we still saw droplets, but we found that increasing the amount of cationic residues actually resulted in dissociation of the droplets. And we actually believe that, for example, at really high concentrations of the cationic residue, it's very likely that the droplets are too polar and too soluble in water, or they're too charged and too soluble in water, so they don't form the droplets. But at lower percentages of the cationic residue, we still see droplets, and it's very likely that these droplets are still cationic. So it's likely that the polyester formation is governed by the total charge of this mixture. And in fact, we did find the ability for these new novel, cationic droplets to segregate RNA. You can see here on the bottom that the fluorescent RNA is really strongly segregated into these droplets here. And we found that even increasing the amount of charged monomers into a neutral polyester by only about 20% allowed polyesters which originally, when in the neutral form, couldn't segregate any RNA to be able to segregate some RNA here shown in the middle and the right. So even just a small amount can change these structures. And so again, if you're interested in learning more details about this study or these studies, some of them have been published. I would highly encourage you to read some of these papers here for more details. So I'd like to, of course, thank my funding sources. A lot of these funding sources came from Japan, but also one from the French Embassy in Japan. I'd like to thank some of my collaborators that have worked on this project with me before, especially Irina, Jim, Kuhan, and Tomazo, both at ELSI and outside of ELSI. And I'd like to thank other collaborators at ELSI and Tokyo Tech, especially Chen and Rihanna, who recently have been working with me on some other really exciting projects related to polyesters. And also a lot of my collaborators around the world for all these projects as well. It's been a great experience, and I hope to continue working with everyone. So finally, I'd like to give a few, programs that you might be interested in to get into astrobiology research. So Blue Marble Space Institute of Science has a young researcher program over the summer. If you're studying in university or recently graduated and are interested to do a virtual project over the summer, you can take a look. There are different projects of different types, and some are research-based, some are outreach-based, some are communication-based. And so you can take a look at the website shown here. My main affiliation, ELSI, we're a research institute located in Tokyo, and we study the origin and evolution of Earth, planets, and life on Earth. And here's some other pictures here. We've recently started a 5-year integrated master's PhD program, and this program is open to both domestic and international applicants, and you will be fully funded. If you're interested, the next deadline for fall 2022 enrollment is mid-April. You should visit this website here. Hopefully you'll be interested in this. And finally, last year, I was part of the organizing team for a conference called AbGradCon, which is Astrobiology Graduate Conference. It's an annual conference for students and postdocs in or looking to get into astrobiology, and it's organized by students and postdocs. And it's a really great experience, especially if you've never done a formal astrobiology or formal presentation. It's a really good experience and opportunity. Graham actually also was one of the other speakers of this session, was an organizer back in the day as well. So the next edition is online. It's going to be held in August, and the abstract deadline is May 15th. So if you're interested, please visit the website and the new team of organizers. They've been doing a really great job, and I highly encourage you to attend that. So thank you all for your time and attention. Thanks again to the organizers for giving me this opportunity. I'm sorry again that I couldn't be there live with you, but I'm very happy to take questions either by email or if you want to leave a comment on the YouTube, I'm sure we can be in touch. So thank you again, and I hope you enjoy the rest of this meeting.